Okay. Okay, so I would like to thank Cedric Chaveroux for taking the time to share his latest results. So he completed a PhD in nutrition at the Clermont University in 2009 in France. And he has several external postdoc stays, and he joined the Cancer Research Institute of Lyon in 2016. So Cedric's research is focused on molecular mechanism linking nutritional stress and cellular plasticity. He's particularly focusing on the response to ribotoxic stress and the contribution of DCN2 in signaling pathway to cell adaptation and translation. So with this introduction, I will give the floor to Cedric. Thanks again, Cedric, for your time.
Well, thank you for this opportunity to present this work. I'm going to try to share again my screen. It should work.
We can see your screen now. It's not in full screen mode, but we can see your presentation.
Yeah, I'm gonna share. Okay, so today I'm going to talk about the latest study that we submitted this week. So it's really recent. It's not published data. It's accessible online if you want on Bureau Archive, but still not published yet. So I'm going to talk about targeting the cell and non-cell autonomous regulation of the ribosome biogenesis by GCN2, and with a particular focus on colon cancer. As you may know, in different contexts, pathophysiological contexts, cells will experience a metabolic stress, meaning that they're going to face a stress with a low availability of nutrients. And this is particularly the case across aging, wound, inflammatory diseases, ischemia, cancer. And actually, the outcome for the cell is first the death, and this is not dramatic. At least there is no particular consequence on the pathology, but it can also induce a plasticity program that, if the stress is moderate and then the cell can handle it, will progress to disease initiation and progression. And actually, the question is how a cell can escape this cell death program to a plasticity program, and see, particularly in cancer, whether we can revert this system to finally kill malignant cells. So we are particularly working in the group on this mechanism promoting cell survival and plasticity upon nutritional scarcity. As you may know, the protein synthesis pathway is the most consuming pathway for ATP, but also nucleotides and amino acids. So briefly, in a situation where the nutrients are abundant, you have a strong consumption of nucleotides and all these things for ribosome biogenesis, translation, and this will, I don't know whether I can show with the mouse, and this trigger the cell division and support cell division. But in a case where the nutrients are low, we're going to have a strong decrease in terms of ATP, nucleotides, and amino acids, and thus, to survive, cells will lower this ribosome biogenesis pathway and also the translational rate, and actually reprogram particularly this translational program for sustaining this plasticity, cell plasticity. And actually, in the group, we are trying to decipher the mechanism allowing the shift toward a program associated to translational reprogramming in scarce situation, and the opposite. So one key pathway allowing this shift is the GCN2 pathway. GCN2 is a kinase that is activated by scarcity when cells are facing nutritional stress. But not only, since the recent years, it has been shown that actually GCN2 is activated by various stresses, so included nutrition stress, that is leading to ribosomal impairment. And in turn, this kinase will phosphorylate its substrate, EIF2 alpha, and trigger the elevation of transcription factor that is called HEF4. HEF4 will enter the nucleus and enhance the transcription of different genes involved in the adaptive response. So classically in the literature, you're going to see that this pathway contributes to cell capacity through a translational repression. Globally, you lower the translational rate. This is the job for phospho-EIF2 alpha. But also, you're going to have a metabolic response that is mainly driven by HEF4. And this has two main objectives. The first one is to preserve the metabolites for ATP, nucleotides, and amino acids in a tolerable level, and also adapt the metabolism for example, the mitochondria, the angiogenesis, oxidative stress response, autophagy, and others. So basically, if I go back to the original scheme that I showed you, this pathway is controlling the adaptive response through a regulation of the translation, but also the pool of nucleotides. But there was no clue about the role of this pathway on the ribosome biogenesis. So we tried to determine whether GCN2 could also play a role in the lowering of the ribosome biogenesis when cells are facing low nutrients. And why focusing on this ribosome biogenesis pathway? Because it's, again, a highly consuming pathway. About 60% of the nucleotides are dedicated to, of the pool of nucleotides, are dedicated to the transcription, and particularly the ribosome RNA transcription. So it consumes a lot of nucleotides within a cell. And we consider, so it's very variable, but still, that 30% of the protein mass, so the proteins are dedicated to riboproteins, so the proteins that are involved in the assembly of ribosomes. In dividing cells, globally in human cells, it's about 4 to 6%, but it's not negligible. So what is the ribosome biogenesis? So I won't be long on that, just to give you the key elements that you need for the understanding of the rest of the presentation. The ribosome is built through three main polymerase activities. So the first one is the transcription of a pre-ribosome RNA by the polymerase 1 that is called 47S, that is then will be processed for generating the different ribo RNA essential for the ribosome assembly. Then you have the 5S that is encoded by the polymerase 3, and also the riboproteins that are mostly encoded by the polymerase 2. And all those guys will be assembled for generating a mature ribosome. But the thing is, if you're impairing this ribosome biogenesis, like in a situation where nutrients are scarce, this leads to the nuclear stress. And this stress could ultimately lead to cell death. Particularly in low nutrients, it is known that scarcity decreases the transcription activity of the RNA POL1 and the ribosome biogenesis by consequence, but also induces the nuclear stress. And there were few clues showing that GCN2 could be involved in this adaptive response, since the loss of GCN2 dysregulates the amount of 5S and also upregulates the amount of p53, that is a marker of nuclear stress. So we question whether GCN2 could be a regulator of this ribosome biogenesis pathway. So to address this question, we used first 3D models to reproduce the heterogeneous perfusion within the tissue. And you have a highly proliferative zone area here at the contact of the medium, and a casein zone where cells are facing metabolic stress in the core of the 3D models. And to validate that GCN2 is indeed essential for those cells experiencing metabolic stress, we use models of colon cancer, so HET116 here, and we just did a live and dead assay. So we treated these spheroids with the GCN2 inhibitor, and as you can see there, just by adding the drug, you selectively kill the cells that are in the core of the spheroids. So This validates that indeed GCN2 is essential for the viability of cells under metabolic stress, also in 3D models. We also did that with tumor rates derived from colon cancer patients, and we have exactly the same result in live and dead. If you add a drug inhibiting GCN2, you selectively kill the cells that are within the core of the tumor rates. So this validates that GCN2 activity is essential for the cell viability in metabolic stress and casein cells. But the question is whether this role of GCN2 can be attributed to the ribosome biogenesis plasticity. So to address that, we need to go back to a simpler model and 2D, particularly 2D culture. And the main question when we do that is to determine whether the starvation we want to apply is really relevant, comparable to the cancer. And actually, we focus on glutamine deprivation. Why glutamine? First of all, because glutamine is limiting in many types of diseases, including Alzheimer's, systemic inflammation, wounding, and cancer, of course. And there is this very nice paper from Kahn's lab showing that in the different area of tumors, so they took the proliferative area compared to the core of the tumors where the cells are starved, and only few nutrients, in particular amino acids, are lowered in this specific area of casein cells. And among those, you see that glutamine is lower in the core compared to the periphery where the cells are perfused. It's lower, but does it change something? Indeed, because in the same paper, they show that just by lowering glutamine amount, you induce a dedifferentiation process, acquisition of casein state, and chemoresistance. So just by reproducing this concentration of glutamine, you can modify the cell state and induce chemoresistance. So we validated that glutamine deprivation plus the emitter of GCN2 indeed triggers cell death. We have a massive cell death when the drug is added in a condition where glutamine is limited. I'm saying that it is limited since we use comparable concentration of glutamine that has been found in the tumor that I showed you. However, if we start for asparagine, a second amino acid that is also low in the core of the tumors, we do not see any change in terms of cell viability. So let's go back to the ribosome biogenesis and see whether this pathway is indeed affected by the drug when cells are starved. So to address that, we use the fibrillary, that is a canonical marker of ribosome biogenesis. And in low glutamine condition, the inhibition of GCN2, as you can see there, is triggering a sort of diffusion of this marker, meaning that the integrity of the nucleolus is broken. Just to give you an idea on the control condition without the drug, you can see that we have approximately like 4 or 5 cells altogether, and this is the size for only one cell when the drug is there. So just to show you that the scale, the size, cell size is also changing upon the GCN2 inhibitor. We also look at canonical markers of nuclear stress, including TP53, and in cells wild type for TP53, upon low glutamine, if we add the drug, we accumulate TP53, which is a reflect of the nuclear stress that you cannot see in the TP53 knockout cells, of course. And when we look at the ribosome biogenesis, so we did northern blot for assessing the generation rate of the 47S, that is this pre-RNA, essential for the ribosome biogenesis. Just by starving those cells, you see that this 47S synthesis is lowered. This is what expected when you starve the cells. But if we add the drug, actually this synthesis is restored and likely underlying the nuclear stress that we observed here. So what I'm showing you is that the GCN2 activity contributes to the repression of the ribosome biogenesis upon metabolic stress and prevents nuclear stress and apoptosis when cells are stressed. Is it consistent with what we can see in human? Because it is really key for the validation of our data. So we went back to the TCGA data, the database for omics in cancer. And actually, in colon cancer particularly, what is really obvious is that if you compare the main pathway that are dysregulated in cancer compared to normal tissue, it's all about ribosome biogenesis, RNA processes, RNA metabolic process, etc., etc. So there is a clear context in colon cancer for dysregulation of the ribosome biogenesis. And to test whether this dysregulation can be associated to a dysregulation in terms of nutrients accessibility, we made a signature based on the riboproteins that are the most dysregulated, plus or minus, in the tumor compared to the normal. So this is the validation of this gene signature. You can see that it is indeed enriched in the tumor compared to the normal tissue, again in colon. And then we tested this signature against other signatures attributed to metabolic stress, and particularly amino acid deprivation. So in the patients with a high degree of riboprotein dysregulation in terms of expression, you can see that we have a strong enrichment of gene signature dedicated to amino acid deprivation here, compared to the patients with a low dysregulation of riboprotein expression. And this is also the same case if we test the gene signature attributed to ATF4, again, the terminal factor of the GCN2 pathway. What is particularly interesting is if we do the same approach with a gene signature related to hypoxia, which is a well-known stress in cancer, we do not see this enrichment. So basically, what I'm showing you is indeed we can actually correlate dysregulation of the ribosome biogenesis in human tumors to an activation of the at least ATF4 activity. So this is the first part of the presentation. What I showed you is that GCN2 can, let's say, contributes to the plasticity of the ribosome biogenesis, while tumor cells, and I mean cells, are exposed to low nutrients secretion. This has been never shown actually, but the thing is we are still in a situation where GCN2 is contributing to cell plasticity. But in fact, what we observed is the opposite. Actually, GCN2 can also contribute to the ribosome biogenesis adaptation when cells go back to a situation where nutrients are abundant. In fact, what we saw is when we added the drug to our cells, even in a condition where glutamine is abundant, we did observe a decrease in terms of cells by, let's say, 30%, independently of the nutrient context. So we validated this in by different approaches. So we first monitored the cell proliferation. And as you can see, cells exposed to the drug are proliferating lower. We also did the same experience in a situation when cells are, when we refresh the medium just to avoid medium exhaustion and then go back to a situation where we have nutritional stress, and we conserve this effect. We validated this result by siRNA approach, just to make sure that it was not a side effect of the drug, and also on primary human cancer cells from colon, where we see that the drug by itself can lower the cell proliferation in a situation where cells are not exposed again to nutritional stress. However, we did the same experiments where we inhibited the pathway downstream GCN2. So the first one was the E3, which is a compound leading to the inhibition of the IF2 alpha phosphorylation. And actually, this compound does not reproduce what we saw with the drugs targeting GCN2. And it is the same thing with siRNA against ATF4. So if we inhibit GCN2 in abundant conditions, we lower the cell proliferation, but this is independent of its canonical pathway. So we question about this new mechanism. In fact, there is a second, we did RNA-seq analysis first from cells knockdown for GCN2, and we observed again a large dysregulation of functions attributed to ribosome biogenesis. We validated this by, again, the same fibrillary immunostaining, and you see that actually the nuclear integrity are also impaired when the drug is added in conditions where cells are proliferating. And we look at the ribosome biogenesis, so the 47S transcription, and again, just by adding the drug without nutritional stress, you lower this synthesis. So what is the mechanism? Actually, GCN2 has a second substrate that is not really studied, actually. There's only one paper showing this connection between GCN2 and this substrate, is the methynyl tRNA synthetase. And there is a paper from King's lab where they show that this factor can localize into the nucleolus and sustain this 47S transcription. So we asked whether GCN2 can impair the translocation, the translocation of this MET-RS into the nucleus. And to test that, we submitted the cells to the inhibitor. And as you can see, just by inhibiting GCN2, again, without nutritional stress, I'm insisting on this, we have a lower concentration of MET-RS into the nucleus. So we impair the translocation of this factor into the nucleus. And if we inhibit this translocation, if we inhibit the concentration of MET-RS into the nucleus, we also mimic what we observed with the drug against GCN2. We have a diffusion of the fibrillary, meaning that we are impairing the nuclear integrity. The consequence of this is an impairment of the translational activity. So by inhibiting GCN2, we lower the incorporation of fluoromycin, meaning that the protein synthesis is impaired. We have an accumulation of autophagy markers, including P62 and LP3. And by using a reporter, fluorescent reporter system for assessing the autophagic flux, we indeed, just by adding, by inhibiting GCN2, we trigger the autophagy in those cells. As you may know, autophagy activation is also a consequence of the inhibition of mTOR, and we have tested this pathway in our settings. And again, inhibiting GCN2 in those cells inhibit the mTOR pathway. So as illustrated there by the phosphorylation of Ss that is down-regulated upon the drug. Same thing for 4-EBP1 phosphorylation, and of course ALK1, which is the factor that once the phosphorylation is impaired, trigger the autophagy flux. In 3D model, we have exactly the same results. We look at the phosphorylation of S6 in this proliferating zone of those spheroids. And as you can see, just by adding the drug, the intensity of the signal is lower at this specific area. So what we show here is that the inhibition of the GCN2 and ribosome biogenesis axis trigger mTORC1 inhibition and autophagy. So this is new data showing that actually GCN2 is not only involved in cell plasticity, but also is sustaining the cell proliferation, but through a different mechanism that is involving this second substrate, the methynyl tRNA synthetase, and the generation of ribosomes. If we inhibit GCN2 in this case, we avoid the translocation of this factor into the nucleus. We block the ribosome biogenesis, leading to an impairment of protein synthesis, mTOR pathway, and we trigger the autophagic flux associated to a lower proliferation rate. So that is a first demonstration that we can impair the ribosome biogenesis just by inhibiting GCN2. In fact, if we go back to cancer in patients, there is already used compounds that are targeting this ribosome biogenesis pathway that are used for chemotherapies and radio sensitization. It also been used for actinic keratosis and psoriasis, and wide applications through the inhibitors of mTOR that are been found in different type of pathologies. So we took two of those, the actinomycin D, that is a transcription inhibitor. So it blocks the transcription of the ribosome biogenesis, so the RNA polymerase 1 and 3, the two main transcription activity in the cell. And what you can see is that the drug, the inhibitor of GCN2 itself, can kill the cells in the similar way to actinomycin D alone. But if we combine the two, so if we let's say aggravate this this inhibition of ribosome biogenesis, we induce a drastic cell death. This is exactly the same result that we get with oxaliplatin, which is a specific chemotherapy that is known now for triggering cell death through the inhibition of ribosome biogenesis. And to convince you about this data, we made a video of those spheroids treated with the oxaliplatin or the inhibitor of GCN2, and this is what you're going to see there. So the spheroids there are treated with the control, the GMISO, the vehicle. This is the GCN2 inhibitor alone, oxaliplatin alone, and combination of the two drugs. And as you can see there, with the drug, the drug against GCN2 will selectively kill the cells within the core of the spheroid. This is the first part of my presentation. Oxaliplatin alone did not kill that much tumor cells at that concentration, but if we combine the two, we have a dramatic cell death, particularly at the periphery of the spheroids, meaning that we are killing in the cells within the spheroid and at the periphery. We did the same experiment with patient-derived tumoroids, and we have exactly the same results. If we combine the drug inhibitor of GCN2 plus oxaliplatin, we have a strong synergistic effect between the two compounds. So GCN2 inhibition dramatically aggravates cell death upon RNA pol1 inhibitors that are already used in clinics. And this will be the take-home message. So what I showed you today is that GCN2 can control the ribosome biogenesis in both compartments and through two different pathways. It is contributing to the repression of this pathway upon starvation, but in the opposite, it stimulates this biogenesis in nutrient-rich condition. Particularly if we focus on the proliferative cells, you seem to stimulate the ribosome biogenesis through the methionine RNA synthesis factor and not its canonical pathway. And targeting this kinase can aggravate cell death upon other stressors of the RNA polymerase 1 that are already used again in clinics. The last conclusion is to be aware about your nutritional milieu, but particularly when you are doing in vitro assay, just to make sure that you are in proliferative state or not exhausting medium. And I would like to thank my group, particularly Marie, who did most of the work that I've presented to you, and all the guys involved in this project. And thank you, and I will be glad to take questions.
Thanks, Cedric. Very nice presentation and very nice videos also. And I like the combination of bioinformatics and experimental data. So very nice presentation. So any questions from the audience? So maybe I can start with the first question. So What about so now with this inhibition of GCN2, how well is the modification characterized upon this inhibition, and is this something of relevance to you?
The mechanism you mean between
I was thinking post-translation modifications, so the role of post-translation modifications, if you think studies like this would be of more interest. And so you mainly focus on phosphorylation. Of course, you talk a bit about phosphorylation, but there was very little about post-translation modifications in your presentation.
Actually, regarding, so we have to distinguish both compartments. In start situation, what we think is we are just targeting the canonical pathway through this phosphorylation of eif2 alpha, and this is well described in the literature. It's clear that GCN2 is phosphorylating eif2 alpha on this serine 51. I mean, this is clear. What is really, let's say, more new is this role in the MET-RS, in the control of MET-RS translocation in proliferating cells. And actually, this has never been showed for me. And the mechanism behind, we don't know. We still know that we are blocking MET-RS in the cytosol through the drug. But we don't, this is what we're working now, actually. We are trying to decipher what is the mechanism by which GCN2 is controlling this, this translocation, and what are the modifications, because still GCN2 is a kinase, so we think that it's going through phosphorylation, different phosphorylation, but we don't know whether it's directly MET-RS or another substrate that is involved in the same complex. So the first thing that we need to determine is what is in the complex and what are the post-translational modifications that are affected by the drug in proliferating state.
Okay. So many years ago, I actually worked with the EF2 phosphorylation, or EF2 and the phosphorylation. But from that I know that there is many stressors that lead to phosphorylation of EF2. So my question is, how specific, or do you think that GCN2 is responsible for most of these phosphorylations, or is there other pathways that can lead to this phosphorylation? So what I'm alluding to here is, of course, the specificity of the system in terms of using it as a drug, in terms of toxicity and so forth.
From what I know, so there is 4 different kinases capable to phosphorylate eif2 alpha. There is a 5th one that has been identified 2 years ago, but there is no much literature on this. So I'm going to talk about only the 4 main kinases that are pretty clear: PKR, PERK, the one that is associated to ear stress, GCN2, and HRI. We use different inhibitors specific to those kinases, or knockdown or extra, and we only see this effect with the inhibitor of GCN2. With siRNA against GCN2 knockout, we have done all the different approaches that are being required, because in the literature, in the community, most of the time GCN2 is just a switch. It's important for stress condition, but there we have an anti-proliferating effect without any metabolic stress. So we are pretty sure that what we have in hand is really specific to GCN2. To be clear on this, I don't think it is specific to cancer cells, because if you take those fibroblasts from, like normal fibroblasts from the mouse that are GCN2 knockout, we do have the same effect, but they are not cancer cells. So we think that it is GCN2 specific, but again, the mechanism behind is, I would say, dependent of MET-RS, but I don't know whether this is something that can cross with other kinases. But for us, the other kinases related to eif2 alpha are not involved in this mechanism particularly.
So there is phosphoprofiling studies inhibiting these 4 kinds, because since they all lead to the same phosphorylation of EF2, but the function seems to be different. So then this should be reflected in the overall profiling. But there are studies that have, are you aware of studies that have done overall phosphoprofiling inhibiting each of these 4 kinases to see the differences? Or I guess that could be interesting to see. in terms of crosstalk in the signaling pathways?
Yeah, for me, you have a lot of data, not really on post-translational modification.
Okay.
On gene expression, yes.
Okay.
So even at the translational level, there is not that much studies on that.
Okay. So you use the TAP20 inhibitors. So are there other inhibitors
so
that you could recommend?
Yeah. So well, the inhibitor that we used was a collaboration that we got with Merck, to be clear.
Okay.
And was coming from Merck. There is a second inhibitor that has been that is commercially available, and that is called GCN2EB.
Yeah.
This is the one that is most of the time used for particularly in vivo approaches. In our hands, this compound is indeed inhibiting this phosphorylation of eIF2 alpha by GCN2 under stress, but we do not see the effect on the matter rest, even in stress cells. So the GCN2B has been specifically designed for inhibiting the GCN2 ISR eIF2 alpha pathway. But if you are interested in the other branch, it won't work. So if you want to distinguish, and actually this is what we're working on, because we do have contexts where we can activate one and not the other branch, or the two branches at the same time. I would rather recommend for in vitro model, the A92. The problem is this is not for us, at least for the regulatory rules, soluble enough for being used in mice. But for the GCN2EB, for us is not particularly, if you want to just focus on the ISR, fine, it works. But if you want to look at the two branches, I would recommend the A92.
Okay, thanks. So Dimitris have a question.
Yes. Hi. Very nice talk. I was wondering regarding the pathway you describe, where you promote when inhibition of GCN2 promotes the controls the phosphorylation of IF2A and potentially protein translation. Independently now of what's happening in the nucleolus, I mean, have you seen changes in cytoplasmic inclusions like stress granules, which IF2A, normally goes there upon stress?
We haven't checked that actually. To be clear, when we found that actually GCN2 is controlling the ribosome biogenesis in stress cells, for us it was quite, let's say, expected in a sense. Mm-hmm. Since there was at least one paper showing that GCN2 was able to, let's say, impair this pathway when cells are upon nutritional stress. And when we saw that actually the same kinase is controlling the same pathway, but in the opposite way in proliferating cells, we focused particularly on this pathway. Mm-hmm. Because it was more original. But you're right. Now, looking at more specifically this literature, I think we have missed a lot of things in stress cells. And I would say there is a large panel of contexts where I think GCN2 is essential for cell survival in different types of stresses, and particularly proteostatic stresses. And they are not relying on eIF2 alpha.
Okay.
We think that what I'm claiming today, what I'm trying to demonstrate is, in fact, this eIF2 alpha is important for cell plasticity, but not essential for cell survival. And we think that the it is the one that is really important.
Okay, thanks. Thank you.
Welcome.
Any other questions from the audience? Maybe if you allow me, I will ask the last question that I have. So normally with inhibition of autophagy, you have it can go both ways, right? You can either you can stimulate cell death either by activating or deactivating, and I think there are also some claims that GCN2 can be targeted in the same way, either activating GCN2 activity or inhibiting it. You were more focused on inhibiting it, but what do you think about the alternative pathway?
I think we have exactly the same thing that in your stress. You have people saying that particularly in cancer or other type of disease, but in cancer it's really let's say debated. You have people saying that we have to activate the stress and kill the cells through the terminal ISR, so the terminal pathway of the EIF2-ATF4 pathway, or you should repress it. In vitro, it works very well to kill cells by adding compounds that are activating GCN2. This is like allofuginine. In vivo, this is not particularly obvious. Because what I think is, in a normal situation of stress, in cancer, but also, again, this is not general to cancer, in other type of diseases, cells are also already facing stresses, and all the feedbacks are set up for avoiding the stress at a level where the apoptosis is induced. And I don't think that over-activating the pathway in vivo will reach this sufficient level of stress for activating apoptosis. So I think that inhibiting the pathway would be better. The main question behind that for me is to determine in which specific context actually the GCN2 pathway can be modulated and will have a really specific effect on cell outcome. So the subgroup of cancer or other diseases where the pathway can be really essential. Because I think there's not just a stress and you have GCN2 activated, you have specific context where GCN2 is indeed activated. And this is where it is relevant to target.
Okay, thanks. So any last question for Cedric? Otherwise, I will again thank Cedric for taking the time for this nice presentation and share your latest result with us. That was very nice. Thanks.
Thank you very much. Bye, guys.
Thank you. Bye-bye.